This video will be on bacterial genetics, the second part. We will briefly talk a bit more about plasmids, and then we'll talk about transformation, followed by transduction. So the fertility factor that we talked about in the previous video was actually one of the very first plasmids that was discovered in bacteria. And since that time, there have been a lot of other plasmids that have been discovered. And so one really common type of plasmid in bacteria are called R plasmids, which carry antibiotic resistance genes. And these plasmids can be transferred very easily between bacterial cells. And this is the reason for the rapid spread of antibiotic resistance in bacteria. The other really important use for plasmids has been in biotechnology, and people have started to use these plasmids as what we call cloning vectors. And so these are commonly used to insert another piece of DNA that you're interested in studying into the plasmid so you can study it in more detail. And so here's an example. You might take a human gene out of a human cell, cut it up with restriction enzyme, and insert that gene into the plasmid. And then very common use has been just to make a lot of copies of the gene that you're interested in studying. So you insert that recombinant DNA, that plasmid with your gene of interest, into bacteria via transformation, and then you grow up a whole bunch of bacteria. And so now you have many, many copies of that plasmid of interest, and you can actually purify that plasmid and get lots and lots of copies of that gene available for study. This was the only way to make a lot of copies of a particular piece of DNA prior to the discovery of polymerase chain reaction. So one really common use in medicine using bacterial plasmids is actually to make human recombinant proteins. And so what people do, for example, individuals with type 1 diabetes need to have supplemental insulin. It used to be they had to purify that insulin from pigs, I believe, and now they actually have something called human recombinant protein that is produced by bacteria from the actual human gene. So if we take out the human insulin gene from a human cell and insert that into a plasmid, so that little yellow piece is the human insulin gene, put it into a plasmid, then transform that plasmid into bacterial cells, you grow up a whole bunch of bacteria, and we can induce those bacteria to actually produce and secrete human insulin. So there are other human disorders that are treated by recombinant human protein, such as hemophilia. You can have the bacteria produce the particular clotting factor that individuals are deficient for, and then inject that into their blood. Okay, so next we're going to move on to talking about transformation. So transformation is the ability of bacteria to take up free DNA from their environment. So bacteria can take up free DNA from their environment. If it's a long strand of DNA, it can actually be used to actually insert into the bacterial chromosome. Or if it's a plasmid DNA, that plasmid will then just be replicated separately from the genome in that bacterial cell. So your book goes into some details about how transformation has been used to study linkage, actually, in the donor DNA. We're not going to go into that much detail about it. I'll just suffice to say that there are two main ways that people use to transform bacteria. One is by the method we used in lab, which is chemical transformation, where we induce the uptake of DNA from the outside of the cell via a heat shock. The other method is via an electrical shock called electroporation. So an experiment we talked about back when we talked about the DNA structure actually was the first observation of transformation. So if you remember, we talked about Griffith's experiments where he took this streptococcus bacteria and injected it into mice. And he found if you actually mixed this heat-killed strain of virulent bacteria together with a non-virulent strain, you would actually kill the mouse and you could isolate virulent cells from that mouse. What was actually going on here was transformation. And so that dead cell extract of the virulent strain included pieces of naked DNA, which were then taken up by the non-virulent strain, and those pieces of DNA changed that non-virulent strain to become virulent. The third way we'll talk about that bacteria can exchange DNA among themselves is called transduction. And this is using an intermediary called a bacteriophage. It's a type of virus that only infects bacteria. So bacteriophages are generally have either an RNA or a DNA genome that are in this protein coat, and when a bacteriophage infects a bacterium, it actually lands on the surface of that cell and will inject its DNA into the cell. What happens then is that that DNA that has been injected can then take over that bacterial cell, and it can result in the production of a lot more viruses, bacteriophages, that will eventually lyse the cell and kill it and spill out those viruses, which can then go on to infect other bacteria. A couple of things can happen upon infection of a bacterium with a bacteriophage. This is a little bit of a complicated slide, but I'd just like to go through it in detail. So here is where the bacterium gets infected with a bacteriophage. And so here is the virus on the surface of the cell. The genome gets injected into the cell, and at this point, there are two possible things that can happen. Let's go to the left first to the lytic cycle. So first possibility is that that genome immediately begins to be transcribed and also copied so that you can make a number of viral genomes and also make the proteins that are important in making that viral particle. So as this happens, it actually begins to kill the bacterial cell. And as a part of the process of killing that bacterial cell, the bacterial genome, which is shown in green here, starts to get fragmented. And so you can see this is getting chopped up into little pieces, and in the process, this cell is starting to assemble these new bacteriophages. And so here we have partially assembled phages, and eventually those are going to kill the cell, and you will have lysis, and more viruses will spread to infect other cells. Okay, that is the lytic cycle. It results in the killing of the bacteria. Another thing that can happen when that viral genome gets inserted into the bacterium is that that viral genome could actually insert into the bacterial genome. When this happens, it is called a prophage, and this puts it into what we call the lysogenic cycle. This bacterium actually inhibits the transcription of those genes in that prophage, and so that prophage just sits there in the genome, sort of in a silent waiting stage. During this period, that bacterium can actually continue to divide, and so it's infected with the virus, but it does not actually get killed by the virus immediately. These bacteria grow, but then at some point, that virus can actually pop back out of the genome and go back and enter into the lytic cycle. There are two types of phages. There are either temperate phages, which can do both of these types of cycles, either the lysogenic or the lytic cycle, or virulent phages, which can only undergo the lytic cycle. Now, how is this process exploited in order for bacteria to share bacterial genes among each other? So there are two ways that this can happen, and we're going to talk about both of them. First of all, generalized transduction. Second, specialized transduction. Generalized transduction always involves the lytic cycle. What happens here is you infect the bacterial cell and you start to kill that bacteria. And during the process of making new bacteriophages, a mistake can actually be made in rare instances. So rather than putting the bacterial genome, which is that purple piece of DNA, into a phage head, sometimes the phages actually make a mistake and take a piece of that bacterial genome that is getting fragmented, which is shown in red in this figure. In this case, you make a bacteriophage that actually does not encode the virus genome. That bacteriophage is perfectly functional as far as infecting another cell, however, and so when that lysis occurs, that phage that had a piece of the bacterial genome can go and infect another bacterium, and it can insert that little piece of bacterial genome into another cell. That piece can then undergo recombination, and you can get sharing of bacterial genes in this manner. This is called generalized transduction because any part of the genome, of that bacterial genome, can be mistakenly inserted into the phage head. In contrast, specialized transduction involves cells that previously underwent lysogeny followed by lysis. And so here you have a prophage, so previously a virus has infected that bacterium and the phage genome inserted into the DNA. And it turns out that each phage has a preferential place that it inserts into the bacterial genome. So there have to be a particular sequence present in order for a particular phage to insert. And so this is really the nature of a virulent versus a temperate phage. Virulent phages do not actually have a insertion site, a preferred insertion site into the bacterial genome, whereas temperate phages do have a place where they can insert. So what happens in specialized transduction is this phage will then pop out of the genome, and if it comes out and takes part of the bacterial genome with it, and what can happen is that certain genes can be packaged into the virus along with the viral genome. When this virus goes and infects another cell, we can actually get sharing of this bacterial gene, the gal gene in this case, with another bacterial cell. And so this one was mutant for the gal gene, and we can actually transduce that wild type gal gene that came from the prophage harboring cell. This is called specialized transduction because since the virus inserts at a particular place in the genome, the same genes are always going to be transferred for that particular virus. So for example, this particular phage always will insert near to the gal gene, and either a gal gene, which is on one side, or possibly the gene that is on the other side of that specific insertion site will always be the gene that is transferred by that particular virus.